Ever know a large family with many children, all of whom are indistinguishable from each other? Unless they are all identical twins, you have not encountered such a family. Non-twin siblings typically have a range of physical differences, from subtle distinctions in features to looking unrelated. Even though they inherited equal chromosomes from the same two parents, the combination of genes is diversified due to crossing over. Crossing over is the exchange of genes between two chromosomes, resulting in non-identical chromatids that comprise the genetic material of gametes, sperm and eggs. This process results in the millions of sperm or eggs that are produced by an organism, each being different from one another. In other words, every single sperm or egg cell in your body is completely unique. Think of it like two traders meeting to exchange their goods, resulting in both leaving with a more diverse collection of wares than they had before. Thanks to this process, living things have high diversity within populations, allowing for better chances of adaptation to changing conditions and survival of the species. Genetic information is stored in chromosomes, a coiled bunch of genes, which are segments of DNA that code for specific functions or features of an organism. Different species have different numbers of chromosomes. We humans have 46. When your body grows or replaces dead skin or hair, your cells duplicate during mitosis, and the 46 chromosomes are copied before the cell splits into two identical daughter cells, each with 46 chromosomes of their own. This works great for making clone cells, but when it comes to reproducing an entire organism, having a diversity of genes on those chromosomes is key. Meiosis is the process by which gamete cells are made. This occurs in two stages, meiosis I and meiosis II. Meiosis I is basically just like mitosis, except for one key step, crossing over. Crossing over occurs during prophase I of meiosis. The 46 chromosomes are duplicated, now 92 total chromosomes, just like during mitosis, but before separating to divide into two daughter cells, they commence in gene swapping. The chromosomes are essentially squashed together, during which time some genes from one are transferred to the other and vice versa. When the cells divide after crossing over has occurred, the 92 chromosomes are distributed in half, with 46 going to each of the two daughter cells. Each daughter cell has a complete number of chromosomes, and now carry different combinations of genes on their chromosomes. Using our trader analogy, the traders have exchanged goods and now carry a diverse mix of each other's supplies. During meiosis II, the 46 chromosomes are not duplicated. Instead, the cell divides into two daughter cells, each with 23 chromosomes. Each chromosome now has a different combination of specific genes due to crossing over in meiosis I, and each gamete represents a completely unique combination of genes. This halving occurs so that if an egg and sperm combine one day during fertilization, the offspring will have a complete set of 46 chromosomes. Because of crossing over, genes can be inherited independently of each other in future generations. This is called independent assortment, and we see this every day in siblings with different combinations of their parents' genes. One might have mom's nose and dad's eyes, and the other might have mom's eyes and dad's nose. Another might have mom's eyes and nose. This is because the traits for nose shape and eye color and eye shape are all different genes that aren't necessarily passed along during meiosis. But it is often possible for some genes to piggyback along with other genes during crossing over, causing a relationship of linked genes. These genes will not split off independently of each other during meiosis II, and will result in the two traits manifesting together in the offspring. For example, red hair and freckles are traits that are produced by the same protein, MC1R, which is coded for by two linked genes. In the example on screen, the red tip of the chromosome in the bottom gamete cell is being passed on separately from the rest of the red chromosome in the cell above it. How do we know which genes are independent and which tend to stick together? The frequency of independent assortment can be determined by measuring the gene locations on the chromosomes. The further apart they are, the more likely they are to be independently assorted in future generations. The following is a map of human chromosomes. The location of different genes is shown as the different bands on each chromosome. The closer they are together, the more likely they are to stick together during crossover and form linked genes. Let's review. Crossing over is the exchange of genes between two chromosomes, resulting in non-identical chromatids that comprise the genetic material of gametes. This process occurs during prophase 1 of meiosis, just prior to chromosome alignment and splitting of the cell. The chromosomes tend to stick together during meiosis, and consequently swap some genes, which results in more genetic possibilities in the gametes produced, and thus in the outcome of future offspring. We can thank crossing over, in great part, for the incredible diversity of characteristics among individuals in a population, and ultimately for the biodiversity of species as well. This effective little trick of swapping information ensures that our genome contains a wide spectrum of traits and abilities to help continue the success of our species.